one gene into our viral vector. In fact, we could go and get the SMN1 gene from anyone and clone that, and it has very high homology. That means it's the same gene between all individuals. So our gene has been cloned from human tissues. We've sequenced it to know that it's the SMN1 gene, the human SMN1 gene that needs to be replaced and increased in SMA patients.
Okay, I think there's a very good question here from Katie Pitson that many families have wondered. The question about was the child getting protein that was needed, the SMN protein that was needed from the mother while the child was in vitro, or so if she got a blood transfusion, would she get the protein she needs again from it?
Well, it's the SMA kids are missing the cells do not have the number of copies or the level of expression of the SMN. So it's not coming from the blood per se. The motor neurons are actually producing the gene product SMN, and this is reduced. The gene product is reduced in those patients. So the SMN to a motor neuron is not coming from the bloodstream. It actually is being produced within the motor neuron itself. And that's what we're trying to do, is to deliver the gene back into the motor neuron itself so that one increases the levels of SMN in that motor neuron, and at least when delivered early in the mouse, one had significant results on correcting the physiology, electrophysiology, and function of that motor neuron.
So Katie also asks, in your honest opinion, what you have seen so far in the lab, would her daughter Avery, who's almost 2.5 years old, now still benefit from the treatment, or is that something that has to be figured out in clinical trials?
So I think there's some new animal models that will hopefully be coming online and being developed will help to answer some of these questions, and then the human clinical trials, and there's a number of things in clinical trial development or moving towards that, including our own program, that may help shed insight into the disease, and when the proper treatment timing will be. The second part of is there.
She just wanted to know if it's something that we're just going to have to figure out in the clinical trial.
There's basic science studies. There's a significant amount of basic research being done within the field. So some of these things can be answered by transgenic models of the disease, new animal models that are being developed, which are really important. We've got the biological side of what does SMN do in a cell? Is it important in every single cell? Is it more important in a motor neuron? What is it really doing in the motor neuron? There's a lot of fundamental questions in basic biology that remain unknown that may help give insight into a lot of these questions, as well as the basic biology and animal model developments that are occurring in the field. So there's a lot of wonderful laboratories working very hard on trying to answer these questions. Some of these questions are going to be answered in human clinical trials, or likely answered in human clinical trials. Other studies are going to be answered by further advances in our basic understanding of spinal muscular atrophy, the mouse models, and the patients that have this disease.
Okay, so she asked a good question here. The Pitson family has raised over $20,000 through one of the programs that is running now, SMA Kids. She wants to know, what would the money from Avi's Race to Cure SMA be used for? Anything specific? And I would like to add to that, future monies coming in.
First off, I think speaking on behalf of my laboratory, the collaborators, and all SMA researchers in general, we are so grateful for for the amount of commitment and fundraising that the families do to support basic biology and translational studies in this disease. And it really has opened up the door very quickly for some major discoveries. Uh, I alluded earlier in our discussions here on the sorts of studies and the costs that are needed as we move our studies forward. The formal safety studies are extremely expensive. We're talking hundreds of thousands of dollars. The production of clinical grade virus is, again, hundreds of thousands of dollars. We also have basic research questions that are also that are being answered in the laboratory, ways of delivering the virus better. So we have a translational program that is moving forward quite nicely, that we're answering questions on the safety right now. As we move those programs forward, more money is necessary to move closer to the human clinical trials and then to be running the human clinical trials. We also have some data in exploring the potential for muscle enhancement. So this is in spinal muscular atrophy, both severe and less severe mouse models, and those studies are early on, but and we're trying to collect information on, is muscle enhancement beneficial or not in various models of SMA. So clearly, there's a tremendous amount of research being done on just understanding of SMA. So where would that money go? It can be slated towards our translation, and that is getting closer to human clinical trials. That is funding the formal safety studies, funding the production, the larger scale productions for human clinical trials. That is the GMP virus. It can also go to those kind of crazy ideas that we have. So, for example, AAV9 in our gene delivery was a crazy idea that some of these things aren't necessarily always funded by the regular NIH grant mechanism. A lot of the fundings and fundraisers that go into SMA laboratories allow for interesting high-risk questions that might not be funded necessarily by traditional mechanisms to really be tested. This is really crucial for the field, and within SMA, I can tell you, has opened the doors to new discoveries, new therapies, and has been pushing the field forward. This is from the understanding to drugs to oligonucleotides to gene therapy to stem cells. Really, we've seen an explosion of exciting data in the field, and this is this is opening the doors to a lot of programs that are starting to demonstrate promise in the disease of moving to human clinical trials. So we're quite excited about what's happening in the SMA field, and really look forward to our program advancing and the many other exciting programs advancing.
So, but specifically on the gene therapy, future monies, we would have a better idea of where to send this money after your meeting, your pre-IND meeting with the FDA, which will determine what programs need to be funded.
That is correct. The FDA is going to give us feedback on our plans and either increase or decrease the number of studies that are required for us to move forward to human clinical trials. Obviously, if they're asking for more studies to prove safety, the cost goes up. Certainly, we want to do the right studies, and we're going to listen and work closely with the Food and Drug Administration to move forward in the most rapid and in the most safe and most cost-efficient mechanism to get to human trials.
Okay, we just a couple more questions here, and I appreciate you answering all of these.
Very happy to.
Okay, so my question is, can you update us on the non-human primate studies and the purpose for doing these studies?
Sure. So, what are we doing in the non-human primates? I alluded to earlier that the non-human primate studies are on normal monkeys. normal non-human primates. So these monkeys do not have SMA. What we're doing is in the non-human primates is to determine, can we deliver the gene via the bloodstream and still target motor neurons efficiently. We've advanced and we're in the process of concluding those studies. The second study is in a normal monkey that has normal levels of SMN. So monkeys have SMN. If we put in our gene therapy that also increases the level of SMN, is that going to be safe and well tolerated? So those are the sorts of studies that we're doing: safety, and then are we targeting the motor neurons?
So just to add on that, do primates have SMA?
We have not seen a non-human primate develop spinal muscular atrophy. That does not mean to say that it does not exist. It just means that we have never seen it, and we have no available method or ability at this time to test our therapy in an SMA monkey.
So they haven't been developed or engineered yet either. Correct?
That's correct.
Okay. So my question is,
I guess I can answer. There's a number. Dr. Lawson's group is developing in making progress on a larger species. So in porcine, they're trying to make SMA pig, transgenic pigs, which will be useful for testing gene therapeutics, oligonucleotides, as well as chemical compounds that may increase SMN. So this model is in development, and they're making good progress. I'm certain that Dr. Lawson will be giving updates on this, but that would be a larger model that could be useful for studying SMA.
So I wanted to ask the question, what kind of impact has the Pepsi grant had on the research, which was clearly done through the efforts of the community itself?
First off, we're very thankful to the community for their support and their votes to the Pepsi Refresh project, and for the funding to come to advance our translational program. Specifically, this has jump-started our process of developing the gene therapy to scale it up for the potential of doing those formal safety studies and for the potential of moving to human clinical trials. This has put more people to put together the pre-IND package, in preparing all of the paperwork and the regulatory documents and pushing forward on those safety studies so that we have a handle. So really it has jump-started a number of projects on this program as far as production and safety testing, as far as the production of the virus, to move forward on these studies. So we're extremely thankful and grateful for this funding. It's moving us along quite nicely.
And last but not least, what are your thoughts on the other therapies in the pipeline for SMA?
You know, I said earlier that SMA has seen just a number of exciting developments really over the past year and in the past 1 to past 2 years. And a lot of these projects have been in the pipeline for a long time, and they're really moving forward, and some new discoveries that have been made. So I see a significant number of therapies that are demonstrating promise. One is, of course, gene therapy. This is being led by us as well as others. Multiple groups are looking at gene therapy. The second studies that are demonstrating significant promise are the oligonucleotide therapies, exon skipping based therapies that are, and exon based skipping therapies that are demonstrating promise, not only in SMA but also muscular dystrophy, and studies that are being advanced. There's also many of the compounds, chemical compounds and drug-based drug therapies are demonstrating promise and are moving forward and closer to human clinical trials. And I think, so I see three pipelines that gene therapy, oligonucleotides, and drugs is showing significant promise for the disease in treating the disease.